Hello friends, welcome to bioassembling. In this session I will discuss about the structure of DNA and its function. Let's start the session. First of all, DNA. DNA or deoxyribonucleic acid is a helically twisted double chain of polyoxyribonucleotide macromolecule and which constitutes the genetic material of all organisms with the exception of ribovirus. That means ribovirus do not contain DNA. Its exception. In prokaryote it occur in the nucleoid and plasmid. In prokaryote DNA occur in the nucleoid and plasmid. But in eukaryote DNA occur in the chromatin of the nucleus. In prokaryote DNA are usually circular in structure, but in eukaryote DNA are usually linear in structure. These are the structure of DNA. DNA is a double-stranded molecule consists of two polynucleotide chain running in opposite direction, and both strands are complementary to each other. Both strands are complementary to each other. The bases are on the inside of the molecule and the two chain are joined together by double hydrogen bond between adenine and thymine and triplet hydrogen bond between cytosine and guanine. Means adenine and thymine contain double hydrogen bond, whereas triplet hydrogen bonds present in cytosine and guanine. These are nitrogenous base. The pairing is very specific, which make the two strand complementary to each other, and it is very specific type of structure, and it make two strands are complementary to each other, and so each strand contain all the required information for the synthesis replication of new copy of its complementary. Means it contain all the information. DNA contain all the information that is required for the synthesis. Synthesis occur through the replication, and replication is a process where new copy of its complementary base pair is generated. First of all, these are the structure of DNA, and you can see these are the structure of DNA. These are double stranded structure. DNA's full form is deoxyribonucleotide, and it contain, when two strands are separated with a diameter of 2 micrometer, and these are the one helix, one nodes of, and this nodes is about 3.4 nanometer of diameter, 3.4 nanometer, and one nitrogenous base, and second is diameter is about 0.34 nanometer. These are the structure how DNA is formed. These are phosphate, you can see these are the phosphate diester bonds are present, and these are guanine is paired with cytosine, adenine is paired with thymine, and you can see these are the guanine. Guanine is a double ring structure. You can see these are the guanine, and guanine has a double ring structure, and adenine are also double ring structure, and cytosine and the thymine are the single ring structure. So these guanine and cytosine are bounded with hydrogen bonds, and adenine and guanine are the purine base, and the cytosine, thymine and uracil are pyrimidine base. Inside the DNA, thymine are present. You can see these are the thymines are present, but in RNA strands only uracil are present, thymines are absent. These are nucleic acid, the principal information of molecule in the cell. Nucleic acid are the principal information molecule in the cell. All the genetic codes are carried out on the nucleic acid. Nucleic acid is a linear polymer of nucleotide. That imply nucleic acid is a linear polymer of nucleotide. Nucleotide is a compound formed by the union of nitrogen base and a pentose sugar and phosphate. Means nucleotide contain three types of compounded: nitrogen base, pentose sugar and phosphate. Nucleotide is formed through the phosphorylation of nucleoside. And the nature of nucleotide is acidic. These are the nucleotide. Nucleotide are the unit structure of nucleic acid. Nucleotide is composed of three components: nitrogenous base, pentose sugar and phosphate. These are phosphate, these are pentose sugar, and these are base, nitrogenous base here, which adenine, thymine and uracil, whatever it present. And you can see these are the hydroxyl groups present inside the DNA. Second hydroxyl groups are absent, so it is called deoxyribonucleotide. But in RNA, both hydroxyl groups are present. Nitrogenous base are of two type: purines, two ring structure, adenine and guanine. Purines are double ring structure, adenine and guanine, and pyrimidines are single ring structure, cytosine, thymine and uracil. They are the building blocks of DNA. First of all, this is this is phosphate group. Here you can see this phosphate group is bonded with hydroxyl group and three oxygen molecule. And these are sugar, pentose sugar, and these you can see that this is carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. And inside carbon 2 you can see these are the structure. O oxygen groups are absent, oxygen are not present, so it is called deoxyribose. These are bases, nitrogenous bases: adenine, thymine, guanine, cytosine. You can see that the structure of adenine is a double member ring structure, and you can see these are the double member ring structure, and guanine also double member ring structure, and thymine you can see single membrane ring structure, and cytosine. Thymine and cytosine are single, and whereas in adenine and guanine are the double ring structure. And you can see these are the adenine nitrogenous base present in 1, 3 and 7th and 9th position. You can see this nitrogenous base present in 1, 3, 7 and 9 position. 1, 3, 7 and 9 position. These are the nitrogen position, and these are the structure of the building blocks of the DNA. When we talk about the linear polymerization of the nucleotide, the nucleic acid are the form of nucleotide polymer. That imply nucleic acid formed by the nucleotide polymer, and nucleotides polymerization together by phosphodiester bond via condensation reaction. Through condensation reaction, nucleotide polymerization occur, and through the phosphodiester bond is formed. The phosphodiester bond is formed between the hydroxyl group of the sugar of one nucleotide. You can see these are the sugar molecule of one nucleotide, and hydroxyl groups of sugar of one nucleotide, and phosphate group of other nucleotide, and these are the phosphate group of other nucleotide, and this is the structure of phosphodiester bond. You can see this is structure of pentose sugar, and these are the hydroxyl group presents, and there is a hydroxyl group is of one nucleotide, and here is a sixth member, and the hydroxyl group is present and the bounded with phosphate group of the other nucleotide. So this is all about the polymerization of nucleotide. These are nucleoside triphosphate structure. And how this formed? First of all, when these molecules of nucleotide is bound with this molecule through the release of a water molecule, and this is from this type of structure of DNA structure. Polymerization of nucleotide. Nucleic acid are the polymer of nucleotide. The nucleotide formed of purine and pyrimidine base linked by phosphorylated sugar. Nucleotide is formed of purine and pyrimidine base linked by phosphorylated sugar, and the base are linked to the pentose sugar of the nucleoside, and these bases are linked by the pentose sugar of the nucleoside. The nucleotide contain one phosphate group linked to the 5 prime carbon of the nucleoside. These dash, you can see these are the dash structure, and this is called prime, 5 prime. How nucleotide form? Nucleotide is formed by nucleoside and phosphate group. Nucleoside is a compound formed by the union of nitrogen base with pentose sugar, and nucleoside is a component of nucleotide, and it is slightly basic in nature, means nucleoside basic in nature. These are the polymerization of nucleotide. Next, I am come to summary. The polymerization of nucleotide to form nucleic acid occur by the condensation reaction by making phosphodiester bond between 5 prime phosphate group of one nucleotide and 3 hydroxyl group of another nucleotide. Through condensation reaction, polymerization of nucleotide occur, and in this process 5 prime of phosphate group of one nucleotide and 3 prime of hydroxyl group of another nucleotide is bound. Polynucleotide chain are the always synthesized in the 5 prime to 3 prime direction with a free nucleotide and being added to 3 prime OH group of the growing chain. That imply polynucleotide chain are always synthesized in the 5 prime to 3 prime direction and free nucleotide added in the 3 prime OH group. These are the complementary base pairing. It is the most important structural feature of the nucleic acid. It connects bases of polynucleotide chain, polynucleotide polymer with the complementary base of other chain. Complementary bases are bounded via double hydrogen bond between adenine and thymine in DNA and adenine and uracil in RNA. Double hydrogen bond is formed between the complementary base and adenine and thymine in DNA, whereas in adenine and uracil in RNA. Triple hydrogen bond formed between the guanine and cytosine, both DNA and RNA. You can see these are the triple bond formed between the guanine and cytosine of both DNA and RNA. These are the base pairing. You can see that the guanine and cytosine structure triple hydrogen bonds are formed, and these are pentose sugar and these are nitrogenous base. And inside thymine, you can see this structure. Thymine double hydrogen bond is formed thymine between thymine and adenine, and these are pentose sugar, these are nitrogenous base, and these are the double ring structure. Guanine and adenine are double ring structure, whereas cytosine and thymine are single ring structure. And how it forms? A double ring structure bond to single ring structure through the hydrogen bond. That imply adenine is bound to thymine through double hydrogen bond, and guanine is bound to cytosine through three hydrogen bond. This is all about the base pairing. Let's come to the type. Type of nucleic acid. Nucleic acid are of main two types. Two types of nucleic acid: deoxyribonucleic acid or DNA. How it called deoxyribonucleic acid? Because oxygen group of second carbon is absent. Oxygen no oxygen molecules are present in deoxyribonucleic acid. And deoxyribonucleic acid are pentose sugar of deoxyribo, no OH at the second position, Bases are purine, adenine and guanine, and pyrimidine and thymine. Pyrimidines are cytosine and thymine. Means deoxyribonucleic acid contain no hydroxyl groups, hydrogen group, oxygen group present in the second position of the pentose sugar, whereas in and deoxyribonucleic acid yani its DNA, it contain purine and pyrimidine. Purines are adenine and guanine, whereas in pyrimidines are cytosine and thymine. And second type of the structure is the RNA, ribonucleic acid. Ribonucleic acid also contain pentose sugar, it's called ribose. And bases are purine, adenine, guanine, and pyrimidines are cytosine and uracil. You can see that oxygen's molecules are present, OH hydroxyl groups are present, and these are here nitrogen's bonds are bound like adenine, guanine, cytosine, and thymine, and these are phosphate groups are formed in the fifth carbon of the pentose sugar, and due to this oxygen groups are present, so it is called ribonucleic acid, and these are ribonucleotide. Here uracil is present, here adenine is present, here guanine is present, and cytosine is present. So it is called AMP, UMP, GMP, and CMP. How DNA is different from RNA? And you can see that the structure of DNA and RNA molecule contain uracil in the, and whereas in DNA molecule usually contain thymine. It usually, you can see that the structure of uracil base, and these are thymine base in DNA and uracil base in RNA. First of all, it usually occur inside the nucleus of some cell. DNA occur usually inside the nucleus of some cell, whereas RNA occur inside the nucleus of most of the cytoplasm. DNA is the genetic material, but RNA is not the genetic material except in certain viruses like reoviruses. It is double stranded, and with exception of some viruses, means DNA molecules are double stranded helical structure, whereas RNA is a single stranded helical structure. DNA contain over million of nucleotide, and RNA contain depending upon the types, RNA contains 70 to 12,000 nucleotide. DNA's quality is fixed for cell, and the quantity of RNA of the cell is variable. Quantity of DNA is fixed for cell, whereas in quantity of RNA of cell is variable. DNA control metabolism and genetic including variation, and RNA control metabolism under the instruction from DNA, and DNA give instruction to RNA to control its genetic information, to control its metabolism. DNA contain purine and pyrimidine equal number, and in RNA no proportional between the purine and pyrimidine, and DNA is long lived, whereas in RNA is a short lived. DNA is long lived, in RNA is short lived. RNA is a nucleic acid polymer that use slightly different sugar than DNA and base the uracil in place of thymine. This all about the difference between the RNA and DNA. And you can see these are the structure of RNA and DNA. Structure of RNA is single stranded, whereas in DNA is a double stranded structure. And you can see there sugar phosphate handles, and bases are guanines, adenine, thymine, uracil are present. Bases are cytosine, guanine, adenine, and uracil in the RNA, whereas in the DNA, cytosine, guanine, adenine, and thymine. The main difference against the structure of DNA and RNA is single stranded. RNA is single stranded, whereas DNA is double stranded. Uracil is present in the RNA, whereas thymine is present in the DNA. So this all about the this structure of DNA and RNA. In our next upcoming lecture, I'll discuss about the function of RNA and how RNA replicate. If you like this session, please like, share and subscribe. Thank you for watching.